full lab today. How about the water away, please? Not yours. Okay. Thank you. All right. Well, let's see. Let me start by recapping a little bit about what we did last time. The idea is that we are pretending we're a research laboratory and we're trying to clone one particular gene out of the thousands of genes that are in this jellyfish. And this is supposed to be one of the chromosomes on the jellyfish. And the name of the gene is the green fluorescent protein gene. And it makes a certain protein which, for reasons that I don't know why, glows green. Maybe the jellyfish uses it to communicate in some way, I don't know. Anyway, for whatever reason, we're interested in that particular gene. And so our approach was this, to isolate genomic DNA from the jellyfish, and then using restriction enzymes, we cut it up into thousands of little fragments, one of which will have our gene of interest. But let me ask that the electronic stuff go away. Thank you. One of those fragments has our gene of interest, but the other ones have the other genes that we're not interested in. And so as of yet, we haven't really sorted through any of these fragments to try to find our gene of interest. But what we did do is take all these fragments and ligate them into plasmid, which has also been cut with the same restriction enzyme. And so we ended up with something called a genomic library. A genomic library is a collection of pieces of DNA ligated into some sort of cloning vector. And a cloning vector is any piece of DNA that you can use as a copy machine. Plasmids count as cloning vectors. So one of these, let's say that one, has our gene of interest, and the other plasmids, well, they have jellyfish DNA, but not the pieces that we're interested in. And in this diagram right here, this shows what we did, when was it? I guess last week. We took the plasmids and we transformed the bacteria with the plasmids, meaning we put these recombinant plasmids into bacteria. And what you guys did after the transformation then was take those bacteria and plate them, which means put them on a petri dish that has some growth media on it. What happened then, so after you put these plasmids into the bacteria and put them on the petri dish, the transformed bacteria landed at different spots on the dish, and you spread them out. But at first you couldn't see any bacteria on there because individual bacteria cells are microscopic. But what happened is Joni put the petri dishes at 37 degrees, and that's the perfect growing temperature for bacteria. And so each bacteria started to grow and grow and grow and grow and grow. And so after around 24 hours, each of those bacteria had grown into millions of copies of itself, so to speak. And you could start to see these things called colonies on the plates, each colony being, I don't know, millions of different bacteria. And this one right here, you can see, I don't know, probably 100 different colonies on it. So each one of those colonies came from just one bacteria originally. And so therefore, all the bacteria in each colony have all the same plasmid. You know, maybe this one has this plasmid, and maybe this one has this plasmid, and maybe this one right here, this colony right here, maybe that's the one that has the plasmid with our gene of interest. So if you were a researcher, your next step here is now what's called screening the library. Screening the library means trying to sort through these various colonies and figure out which one has your gene of interest. And you know, as I've drawn it here, it only looks like there's just a few colonies, maybe 7 or 8, and the same on this diagram right here. You might think, well, that doesn't sound too hard. Just sort through a few colonies and I'll find which one has my gene of interest. But you know, in real life, this would actually represent tens of thousands or hundreds of thousands of different plasmids, and so it's not a trivial task to look through all those colonies to figure out which bacteria, which colony has your particular gene of interest in it, in its plasmid. You know, in a way, it's sort of like sorting through a needle with a haystack. And so what I want to talk about here is from your DNA technology lecture. So if you got those notes, take them out. It's how you screen a library, how you find your particular clone gene of interest. So the first part, I guess I spelled out here, you make recombinant plasmids and you transform bacteria with those plasmids, and then you plate them out on agar plates that have the growth media on there, and then each bacteria grows into a colony of bacteria, a clump with millions of bacteria. But each colony originally came from one single bacteria, so each colony all has the same plasmid. But now we have to sort through and find out which plasmid has our gene of interest. Okay, well, to begin screening the library, what you normally have is something called a probe, or what you normally use is something called a probe. And here we go. Suddenly I see that I don't have an overhead that I needed. Give me 1 second to see if I can dig out that overhead, and then I will continue. Here we go. All right, so this probe is a tool that you're going to use to find out which colony has your gene of interest. And the probe is either a small piece of DNA, or I should have said RNA here. It doesn't make any sense. It's a piece of DNA or DNA. That sounds brilliant. It's a small piece of DNA or a small piece of RNA that's complementary to your gene of interest. In other words, if this is a sequence from your gene of interest, a probe is a small piece of DNA or RNA that's complementary to part of your gene of interest. You can see it has the complementary sequence there. And the reason why it's supposed to be complementary to your gene of interest is, remember this amazing property of nucleic acids. If two pieces of nucleic acid are complementary in sequence, they can find each other, like a dog that can find its way home from hundreds of miles away. This probe has a natural ability to stick to just this piece of DNA, just your gene of interest, because it's complementary in sequence. Now, you may say, well, where do I get this probe from? You can order probes from biotechnology supplying company. You can tell them, hey, I need a probe and it has to be the sequence CCGATTGAATCG. So the problem is not really getting the piece of DNA, it's knowing which sequence it's supposed to be, because to know which sequence your probe should be, you have to know what the sequence of your gene of interest is, and you haven't cloned it yet. You know, you're still in the process of cloning it, and so how could you possibly find the sequence if you haven't even cloned the gene yet? So in a way, it seems like a chicken and an egg paradox. You can't find it until you have the probe, and you can't make the probe until you found the sequence already. Well, there are workarounds. Even if you haven't cloned the gene yet, oftentimes researchers have done a little bit of research on the protein, on the gene product. And if you have some of the protein, you can look at the amino acid sequence of the protein and guess some of the codons that are likely to be in the gene. And oftentimes that's good enough to make a probe from. And another workaround is this. If you're studying a certain gene in one species, it could be that another researcher has already cloned the gene in a related species. You know, if this is the GFP protein from jellyfish, maybe someone who's studying squids or octopus or whatever related organism is might have already cloned the gene. And you know, they're not the same species, so the sequence of the gene might not be exactly the same, but if it's close, you can base a probe on some other species and it will probably still work. Anyway, so the major point here is that you need to get a probe that's complementary to your gene of interest, or at least very close to it in the sequence. And typically the probe is also made out of radioactive nucleotides. And the reason why that is, that's going to be a sort of a signal. Once your probe binds to your gene of interest, by emitting radioactivity, it's going to say, hey, I found the gene of interest and it's right here. It's on this particular colony. Okay, so let's watch the process here. You transform bacteria with the plasmids from your genomic library. You've grown them up on a plate and they've grown into colonies. And so the question is, which colony has your gene of interest? Let's see, here we go. What you do is this, you take the plate that you're screening, and on top of all the colonies, just right on top of the plate, you add what's called a filter, which is just a fancy term for a piece of paper or a piece of nylon. And as you lay this piece of filter on top of your plate of bacterial colonies, just by the sheer weight of the paper, which is not much, a few of the bacteria are going to be lysed, are going to be broken open, and that means some of their plasmid DNA is going to transfer over to the piece of filter paper. Not all of the bacteria will break, but at least some will. So now, although you can't see it immediately, on your piece of filter are small bits of your plasmids from this colony and this colony and this colony and this colony and this colony. It's almost like an image of the plate has been transferred to the piece of filter. Once you have that, once you have the filter, you hybridize the probe to the filter. And hybridize just means to allow complementary nucleic acids to a complementary base pair to each other. So in other words, you just soak your filter in a liquid that has the radioactive probe that's complementary to your gene of interest. And remember that principle, a nucleic acid will be able to find just the piece of DNA that it's complementary to and stick to it. So it will stick to your gene of interest and not to any of the other colonies that have different genes on them. And so some of these colonies are then going to get radioactive. Whichever ones have your gene of interest get radioactive because the probe is sticking to those colonies. Now, we can't see radioactivity with our eyes, so we need some way of visualizing them. And that's pretty straightforward. Photographic film can be exposed by radioactivity. And so the filter that has the probe hybridized to it is put in contact with a piece of photographic film, and then areas of that film that correspond to the areas of the filter that are radioactive will get exposed. And when you develop this, you'll see little dark spots there and there, indicating that the colonies in those positions on your plate are the ones that had your gene of interest because the radioactive probe stuck to them. And so you simply then go back to your original bacterial plate and say, okay, line up the film with the plate and say, where are those two colonies that my probe bound to, this one and this one, and so you know then that those colonies contain the plasmid for your gene of interest. So you've essentially sorted through hundreds or even thousands of colonies and found the ones that have your gene.
Why is the DNA single-stranded?
Why is the DNA single-stranded? Oh, you mean right here?
Yeah.
Oh, I see. That's a good question. Let me run it backwards slightly. Go back. Here we go. When you had your piece of filter paper that you put on the colonies, before you hybridize it, you treat it with some things like sodium hydroxide that denature the DNA, meaning make the double-stranded single-stranded. So you've converted it to single-stranded before you hybridize it with the probe. Okay, yeah, excellent question. I can see you're following along. Okay, so Anyway, so when you've gone through this procedure, you end up identifying which colonies on your plate are the ones that have your gene of interest. And so where do you go from there? Well, typically when people have found the ones that have the plasmid that have their gene of interest, the next thing they might want to do is sequence the gene, because by sequencing it you can learn about the exon structure and the intron structure and the alternative splicing. And if you look at the promoter region, you might learn a little bit about the expression, what regulates the expression of it. So you'll essentially need to grow up these colonies to larger amounts, because the amount of plasmid that's in this colony and well, in this colony and this colony is not enough to do DNA sequencing on. You need gobs of DNA for DNA sequencing. So how do you grow up these ones that have your gene of interest? Well, it's pretty simple. If bacteria can do anything, it's grow. And so what you can just do is get a sterile toothpick and grab a little bit of the bacteria from there and then drop them into a flask containing liquid growth media. And so now you've got some of your bacteria that have your gene of interest on their plasmids, and you put this at 37 degrees, and within 24 hours, the colony that has the plasmid that has your gene of interest has grown into this thick liquid. It's just turbid with bacterial